So we're learning about the life cycle of HIV. HIV-1 infects T cells that carry the CD4 antigen on their surface. In addition, certain HIV strains will infect monocytes and other cells that have CD4 on their surface. Binding to CD4 on cell surface and viral entry is also mediated by chemokine co-receptors CXCR4 or fusin and CCR4 R5 present on T cell and monocytes respectively. After the virus has entered the cell, the RNA genome of the virus is severe transcribed and reverse transcribed, and a cDNA provirus integrates into the host genome. The integrated provirus is transcribed and translated into proteins, which along with a complete new copy of RNA genome are used to form new viral particles. CD4 HIV beta chemokine receptor CCR5, then macrophage, then alpha chemokine receptor CXCR4 C4 CD4 T cell receptor and co-receptors for HIV. Infection of target cell. HIV GP120 binds to CD4 on target cell. Fusogenic domain in GP41 and CXCR4, a G protein linked receptor in the target cell membrane, mediates fusion. Then nucleocapsid containing viral genome and enzyme enters cells. Viral genome and enzymes are released following removal of core proteins. Viral reverse transcriptase catalyzes reverse transcription of ssRNA, forming RNA DNA hybrid. Original DNA template is partially degraded by ribonuclease H, followed by synthesis of second DNA strand to yield HIV dsDNA. The viral dsDNA is then translocated to the nucleus and integrated randomly into the host chromosomal DNA by the viral integrase enzyme. Then activation of provirus. Transcription factors stimulate transcription of proviral DNA into genomic ssRNA, and after processing several mRNAs. Viral RNA is exported to cytoplasm. Host cell ribosome catalyzes synthesis of viral precursor proteins. Viral proteases cleaves precursors into viral protein. HIV ssRNA and protein assembled into which GP41 and GP120 are inserted. The membrane buds out, forming the viral envelope. Released viral particles complete maturation. Incorporated precursor proteins are cleaved by viral protease present in viral particles. Then pathogenesis. Once a human CD4 plus cells are infected with HIV-1, 4 types of pathological changes may ensue. First, a mild form of AIDS may develop with symptoms that include fever, malaise, headache, macular rash, weight loss, lymph node enlargement, oral candidiasis, and the presence of antibodies to HIV-1. This is known as AIDS-related complex. Then second, the mean interval between HIV infection and the onset of AIDS appears to be about 8 to 10 years, although it varies considerably with each individual. At first, a person's immune system responds to the HIV-1 infection by manufacturing HIV-1 antibiotics, but not in sufficient quantities to stop the viral attack. The virus becomes established within primary CD4 plus T helper cells and accumulates in lymphoid organs in large quantities even before symptoms appear. Initially, CD4 plus T helper cells proliferate abnormally in the lymph nodes. Thereafter, the lymph nodes' internal structure collapses due to viral replication. This results in selective depletion of the CD4 plus T cells. When the CD4 plus population declines, interleukin 2 IL-2 production also decreases. Because IL-2 stimulates the production of T cells in general, the whole T cell population may decline. Second, the mean interval between HIV infection and the onset of AIDS appears to be about 8 to 10 years, although it varies considerably with each individual. At first, a person's immune system responds to the HIV-1 infection by manufacturing HIV-1 antibodies, but not in sufficient quantities to stop the viral attack. The virus becomes established within primary CD4 plus T helper cells and accumulates in the lymphoid organs in large quantities even before symptoms appear. Initially, CD4 plus T helper cells proliferate abnormally in the lymph nodes. Thereafter, the lymph node's internal structure collapses due to viral replication. This results in selective depletion of CD4 plus T cells. When this CD4 plus population declines, interleukin 2 production also decreases. Because IL-2 stimulates the production of T cells in general, the whole T cell population may decline. HIV may reduce the immune response by destroying or disabling dendritic cells which present foreign antigens to T cells. HIV may disrupt the balance between the different types of T helper cells and consequently decreases the killer T cell population. In 1993, Center for Disease Control and Prevention (CDC) revised its definition for AIDS to include all HIV infected persons who have fewer than 200 CD4 plus T cells per ml. Healthy individuals have about 1000 such cells in every ml of blood. The decrease in CD4 plus T cells is the hallmark of AIDS. Third, it involves the central nervous system. The classical example symptoms of central nervous system disease in AIDS patients are headaches, fever, subtle cognitive changes, abnormal reflexes, and ataxia. The fourth result of HIV infection is cancer. Individuals infected with HIV-1 have an increased risk of 3 types of tumors: Kaposi sarcoma, carcinomas of the mouth and rectum, and B-cell lymphomas or lymphoproliferative disorders. It seems likely that the depression of the initial immune response enables secondary tumor causing agent to initiate the cancers. It has recently been discovered that Kaposi sarcoma associated herpes virus KSHV, also known as human herpes virus 8 or HHV3, is a virus that is consistently present in Kaposi sarcoma and in primary effusion lymphomas. These cancers occur most frequently in AIDS patients. HIV transmission is transmitted by both homosexual and heterosexual contact, by blood and blood products, and by infected mothers to infants, either partially or via breast milk. Perinatally or via breast milk. There is no evidence that HIV is transmitted by casual contact or that the virus can be spread by insects such as mosquito bite. Similarly, airborne transmission has never been observed to cause infection. Therapeutic agents. At present, there is no complete cure for AIDS. Primary treatment is effective at reducing the viral load and disease symptoms.